So welcome. Just to start out with, I did not choose this picture. I hate Brussels sprouts. And anybody who wants to have the stuffing can have that as well. The rest is fine with me, but it is actually a good picture because it shows a lot of the things that we're eating. And food is very important, not just in our institute, but in pretty much our whole life. Now, food poisoning is very much in the news. These are a couple of cuttings from different years and everything. The most recent one is the one from Lancashire. You may have heard on the news. People were told to boil their water because there's Clostridium in the pipes. And a lot of people were complaining, saying even the tourists won't come back to Lancashire if they have to boil their water while visiting here. But was the advantage, if you boil it at least, that you don't get food poisoning or illness. So it's whatever it's worth it. Similar one, I always like to include the one with the Queen. It's not anything to do with your standing. The whole society can get it. Because the Queen had to cancel a trip to Swansea with gastroenteritis. Of course, it's the Queen, so we'll never find out what she really had. As somebody interested in food poisoning, I'm very interested. Was it Salmonella? Was it Campylobacter? Was it something else? We won't be told. The one at the bottom right here is very recent. That's from this week. This is the CEO from a company in the States who's been sentenced to 28 years in prison. He was selling peanut butter which he knew was contaminated with Salmonella. He falsified the record so he could still sell it and not make a loss. And his brother, I think, got 20 years for this because he was aiding and abetting. But clearly, you get more and more problems now. If you sell food which you know is contaminated, you may get litigation, you may go to jail. So we really need to get things sorted out with food poisoning. And it's, as I say, it's not a UK problem only. The UK has a lot of it. Any developed country has it, and pretty much every country in the world. These are, again, a couple of things. The one on the right top, I very much like the Barre blog. Anybody can go on the internet and find all the horror stories you want. Also, the one I really liked here, that's why I took this one, the tea towel police. Probably the most filthy thing in your kitchen may be your tea towel. How often is it being replaced? How often is it being washed? And say the questionnaire they did with 1,551 people, they were not only that the kitchens were filthy, but the household chefs were also woefully ignorant about food preparation. Now, if you look back in your own, I see that many people here are not the youngest anymore, so you probably have been taught how to prepare your food properly. I wonder whether that's happening nowadays. But wherever you go, say the Listeria in ice cream is not a very good one. And why is Listeria really a problem? Because especially the elderly are very much at risk with that, and the very young. And this is a slide, I need to update it, but the numbers haven't really changed. So this one goes to 2007. But the estimate is that there are about, and this one 765,000, probably 1 million, maybe even 2 million cases of food poisoning in the UK per year. Which leads to about 17,000 hospitalizations, about 500 deaths, and an enormous cost to the UK economy. And if George Osborne would get 2 billion for free to spend on something else, he would very much like that. It can be either infections, and I'll go through a few of them during the talk, or it could be toxins. Now, what I'm not going to talk about now, I'm only going to talk about things that have to do with bacteria. You can also have food poisoning from chemicals. You can have food adulteration. You can have food fraud, or what we call food authenticity, like the horse meat scandal. That is not necessarily a safety issue, but also not something that I work on. But we have other people in the institute who work on food authenticity, where the horse meat scandal really gave them a chance to develop new things. So there are loads of possibilities there. But as you can see, this is really, as you think, an enormous amount of cases and money lost. And if you think about it, what is food safety? Well, nowadays the politicians are very much talking about food security. A lot of people think, well, let's make sure that we have a sustainable production of nutritious food. So is there enough food for everybody to go around? However, there's no point if you make a lot of food if it's not safe. So food security is not just the food, producing enough food, but also making sure that it's safe food, so it can be eaten without making people ill. I don't know whether people know this, but as you can see from the graph, the calorie intake per day in the UK is the 6th highest in the world. I'm afraid that I show a little bit sign of going native as well. The Netherlands is not on this graph, so that's fine. But you can see, I mean, that the USA is top probably won't surprise anybody. But you see Austria, I've gone to Austria for skiing. They have a lot of really nice sugary dishes. Italy, pasta, lots of sugar again. So as you can see, the UK has a lot of food. Our food consumption is very high, but that puts a lot of strain not just on the production of food, but also on the transport, the handling, the retail. Everything has to be right, otherwise we get food poisoning. So what are the numbers then? People like statistics. I prefer the lies, damn lies and statistics version. And looking on the internet actually, so for who does it come from? The Americans claim it's Mark Twain. The British claim is Benjamin Disraeli. I don't know who's right. But say that the UK houses about 65 million people. I don't know whether that's the correct number nowadays, but it's good enough. Say that 60 million of these eats 3 meals a day. And I'm excluding the snacks and the drinks and everything, just the meals. Calculate that's about 71 billion meals per year in the UK. Okay, we said there was about say that we underestimated, say it's 2 million cases of food poisoning a year. That means that you have a 0.003% of the food consumption as actually leading to food poisoning. That's a very low number. So if you think about it, UK food is safe. Now that is something that we really should not underestimate. All these horror stories I'm telling are not saying that our food is not safe. It's just not safe enough. We need safer foods. If we go to other countries, like developing countries, where you have a lot, the irrigation problems and everything, then you would probably get that the food is not safe. But the UK has very safe food. You just need to do even better. So, what is bacterial food poisoning? Well, as I said, there's two different kinds, really. There's the intoxication. So the bacteria are not necessarily there anymore, but they left the toxin behind. You eat the toxin, you get ill. There's the botulism, which is a very good example of it. The bacteria may long be dead. They left the toxin behind, and the botulinum toxin is Botox, which is very well known in the cosmetic industry. It's the strongest poison known. If that's left behind in your food, you can easily die. Another one, for instance, Bacillus cereus, which is why you shouldn't reheat rice. That's one of the causes where you can get Bacillus cereus from. And again, it leaves a toxin behind. The bacteria may not be there anymore. The alternative is the contamination with the bacteria. E. coli is a very well-known one. Everybody's heard of Salmonella. So the bacteria is still there. The bacteria may survive on the meat, but not necessarily grow. If the food is not properly handled or prepared, you eat it, they get into your intestine, they start growing and make you ill. So I thought maybe we'll make it a little bit of a show. I didn't know the podium, so it may not be as easy to play it. But if you think about the pointless show, I can't be as smug as Alexander Armstrong. But is there anybody who would dare to guess? There's an easy one in there, and there's a couple more difficult ones. There's a couple of people who know their food poisoning. Very well done. So, it's the one. If we think about it, I worked with the BBC on a radio interview for Face the Facts, Radio 4, a couple of years ago. They went out in the street and just asked people, do you know any bacteria that cause food poisoning? Not a problem with E. coli, not a problem with Salmonella. Pretty much everybody knew those. Listeria was already very rare, and Campylobacter was not mentioned at all. And that's almost like inverse to the frequency of of the chance of getting these bacteria. And if you would do I don't have any fancy animation with it. If you would do Salmonella, that probably would be the worst answer to choose, because everybody would know that. With Campylobacter or Listeria, you have a chance to score in that show. So this is actually from the government website. So what are the main sources of food poisoning in the UK? I say Campylobacter is the most common one. It's from, the majority is from raw or undercooked meat, but it can also be unpasteurized milk or untreated water, and some more, but those are the major ones. Salmonella, again, raw and undercooked meat, especially eggs, the cookie dough and everything that people would like to have before it's cooked. Milk, other dairy products, pork, there's lots of causes for Salmonella. Listeria is chilled or ready-to-eat foods, pre-packed sandwiches, and I just had one, so I'm just hoping. Cooked sliced meats and pate, soft cheeses are in the news at the moment for Listeria. E. coli, very well known, especially since 2011 in Germany. Undercooked beef, there was the hamburger bug, as they call it quite often, or the McDonald's bug in the States. Mince, burgers, meatballs, again, unpasteurized milk, and there's still people who like to drink unpasteurized milk. I don't know why. Seriously, you're just asking for it. And in the States, actually, people are fighting for being allowed to give unpasteurized milk to children at school because it's healthier, they say. Fair enough. Shigella is mostly from contaminated water. And of course, there are the viruses. Norovirus is in the news regularly when another hospital wing has to be shut down, no visitors because there's an outbreak of norovirus. Or worse, you're on a cruise ship in the Caribbean. And I think that was not this summer, but summer last year, when there was a cruise ship that had to be weeks in the Caribbean with norovirus on board and not enough toilet paper. Parasites, the cryptosporidiosis, the entamoeba and so on, different parasites. We don't work on those. We basically work on the major bacteria causing food poisoning. So how do you get it? I really like that picture on the right, which is sort of like the UV spy cam for where you could find, you think that's a very clean kitchen. And if you then would look where the bacteria are, you see there's a lot of bacteria on the different food preparation places, the chopping board, the kitchen utensils, on the hands. So where do you get it from? For instance, it could be contaminated meat or produce. Your lettuce has been washed with contaminated water, or there were animals walking around defecated on the vegetables. They weren't washed and treated properly. So it could be fecal or soil contamination. That's the first stage. It could be during handling. So it could be improperly stored or handled in the factory, in the shop, or at home. Improperly prepared, again, in the factory, the shop, the restaurant, you name it, wherever they prepare food. It could be cross-contamination at home, from the chopping board to kitchen utensils. And the FSA has been very active with don't wash your chicken. I gather that people have been taught in the past to wash their chicken. What you're actually doing is, because the bacteria are on the skin of the chicken, when you're washing it, you get water drops with bacteria going all over your kitchen. So there's much more chance of getting infection from that. Where you think you cleaned your chicken, you've actually contaminated all kinds of things in your kitchen. It's definitely no-go. Problem is, do people accept that? I told my mum, she knows I work in food safety, she's already had problems accepting it. I've always washed chicken. One of my aunts basically said, I've done it all my life, washing chicken is good. Yes, I tell you it's not, we know now. No, it's good. And I think there will be a lot of people like that who, even with the government advice, may still keep on washing their chickens. Whereas it's definitely a no-go. And then, after all these possibilities already contaminated, if you then don't prepare it properly, undercooking it, for instance, you're done for. You run the risk of getting food poisoning. Now, I said I will discuss briefly some of the perpetrators. E. coli. E. coli was in 2011, there was an outbreak of E. coli in Germany. I think most people will have had it in the news. They blamed the cucumber from Spain. So you couldn't sell cucumber anymore. Nobody would touch cucumbers anymore in the shops, even whether they were from Spain or not. It wasn't a cucumber, and in the end it actually was bean sprouts from Egypt. But the damage had been done, so just a wrong announcement already has an enormous effect. So E. coli is present in the gut of cows and other farm animals. Very important is it does not make the animals ill, so you don't see on the outside of the animal that it has E. coli or not. You can't judge that. The manure contaminates vegetables, but also there could be direct contact with animals. You see a picture at the bottom. There were problems with a petting zoo, where children got ill from petting the cows at a children's farm. They may have patted the wrong parts of the cow, then brought it to their mouth, as children do. They're already a weak one, and once the E. coli is in the human host, it starts growing, it may start making toxins and everything, you get ill. And the German case was really a bad one because a lot of people died from it or had lifelong renal problems or other health problems associated with it. So once you have it, it's not just a case of diarrhea, fine. You may have lifelong problems after that. Salmonella. It's the second most common cause of food poisoning in the UK, but it is probably the most dangerous one. It causes the most deaths with food poisoning. Estimated about in the UK, 120 people per year die from Salmonella. It can come from lots of things: poultry, eggs, pork, beef, vegetables. And we see over the years that Salmonella has become better at actually attaching to vegetable surfaces, so they're now sticking to vegetables even easier, and if you then eat them raw, again you get Salmonella. And this one from the Metro, where people have been getting Salmonella after kissing and cuddling their chickens. Now, again, I don't know what people do in their homes, and I'm not criticizing them, but it is asking for trouble. However, I put this picture on because this happens in my house as well. This is not a picture of me, but I have a cat which comes up to me and shows her appreciation for the food I gave by licking my nose. I know where that tongue has been, I've seen it. So I probably shouldn't allow it, but this happens in a lot of people. I assume quite a few people here have pets. They may have dogs who are licking their face. There is a risk of getting any infection from that. Again, it's your own choice whether you allow it or not. I like my cats, and I'm just hoping they like me also, and not enough to give me some food poisoning or other infections. But the tip of my nose is not the safest position on my body either. Listeria. I've already showed this graph at the bottom of the age distribution of the people who get Listeria and have problems with it, from it. Listeria is pretty much everywhere. It's found in soil, water, vegetation, sewage, and in the features of humans and animals. In factories, we've been working with people from New Zealand on Listeria from mussel factories. And one of the things that Listeria is special in is that most bacteria will not grow at refrigeration temperatures. So you refrigerate it, at least the bacteria won't multiply. Listeria can survive, and they can grow if it's stored in the refrigerator. So you buy it, if it's already contaminated, there may be much more Listeria if you leave it for a few days in the refrigerator. And you won't notice it, there's no effect on the smell or taste of the food. So you don't know that you've been eating it. If you do proper cooking procedures like pasteurization, preparation, fine. And again, things like pasteurized milk, can have a cheese, deli meats. Listeria is deemed the second most important target of the Food Standards Agency, above Salmonella. Not because of the incidence, because it's relatively rare in the UK. However, the people who do get it, there's significant mortality with the people who get Listeria. So, and because it's mostly the weaker groups in society, so the elderly, the very young, people who are immunocompromised, for instance, people who are on immunosuppressive medication, they are really at risk from Listeria. So it's not the incidence, but it's the severity of the disease, which makes it very important. Clostridium botulinum, botulism, basically it produces the neurotoxin, so the Botox, which is the strongest biological toxin known on Earth. Basically, the bacteria are there, they form spores, they're anaerobic, and these spores are very heat resistant. So you have to, when you see here, for instance, an example from hazelnut yogurt. The canned food should have had the botulinum cook, which is 121 degrees Celsius for 3 minutes. It wasn't done correctly to the conserve. The spore survived. So there was growth of the spore, of the germination of the spores in the can, and there was toxin production. And when this conserve was added to correctly chilled yogurt, these people got ill from it. They got botulism. And why is that a problem? I'll say it's the most potent toxin known. And I like this one always. It's 1 million times more potent than cyanide, and 400 grams has been estimated to be enough to kill the whole world population. So my colleague Mike Peck, who leads the work on botulism, he usually has like a bit of sugar, a pot of sugar with 400 grams in it. And I've been told that he dropped it once accidentally during a presentation, and you heard a gasp in the audience, even though everybody knew it was sugar. And you can get it from consuming a few micrograms of contaminated food, and then you may be done for. So, risky. And then finally, the one I'm spending most of my time on, Campylobacter. That's the most common cause of food poisoning in the UK, Europe, USA. It's a bacterium, which you can see here on the right in the picture. Basically, Campylobacter means curved rod. You see a curved rod, it's a bit like a fusilli, sometimes like a spring bean, and then it has these two flagella, these little these hairs that allow it to move. You find it on about 70% of chicken at retail. And the estimates widely vary, because there are about 60,000 cases per year in the UK where there's a proper confirmation that these people had Campylobacter. However, a lot of people, because the signs of the disease are not so severe, they will not go to their GP, or if they go to their GP, there's no fecal sample taken. So there's no official confirmation whether there was Campylobacter there. So we have to estimate it. And the estimates in the EU, because this is not a UK problem, this is EU-wide, is that there are between 2 and 20 million cases per year of Campylobacter food poisoning in the European Union, of the 27 member countries when they did it at the time. If we leave the EU, that's not going to go less. They just lose the contribution of the UK. Now, just getting diarrhea is very unpleasant, and I've been told it's extremely unpleasant diarrhea, but you won't die from it. But you do have the risk of developing paralysis. So as I say, this is a comparison of the number of cases. So it's about 60,000 per year in the UK, but you have to multiply this by, well, anywhere between 4 and 9 times, depending on who you believe. And the estimate of the FSA is about 280,000 cases per year in the UK. It's probably closer to 500,000. The blue one in the same graph is the number of cases of Salmonella. You see the Salmonella cases are going down from 97 onwards. That's when they introduced the vaccination in eggs against Salmonella enteritidis. So they were able to reduce the number of cases of Salmonella. All the things they've tried so far with Campylobacter have not worked. And it's almost like compulsory if you do a Campylobacter talk that you have to show an iceberg picture, because we only know the one case that is laboratory confirmed, but there's a certain number between 4 and 9 that we miss out on. But these are a couple of the major problem makers. Problems are increasing. Things are getting worse, which is good for us, but there's more work, more interest. But pathogenic bacteria are learning new tricks. The outbreak in E. coli was an example of an E. coli strain that hadn't caused much problem before. It learned a few new tricks and suddenly it became this deadly killer. Antibiotic resistance, you probably can't get around it. You must have heard it on the news. We're running out of antibiotics. Resistance against them is increasing. There's now superbugs in the hospital like MRSA, which is pretty much resistant to any known antibiotic. And there's a problem that new antibiotics are not forthcoming. If all these pathogens become resistant to antibiotics, we can't treat anymore. So prevention becomes even more important. The other thing is food production, preparation, consumption, the patterns are changing. That could be national, it could be international. This is work from a picture from a paper from Joseph Barre from our institute, a mathematician. And he started looking at the size of food import and export of different countries. As you can see, Denmark, DNK, Netherlands and Belgium are enormous. You think about it, these are all small countries. So why do they have such an enormous food import and export size? Because of their harbors. Belgium has the Antwerp harbor, Netherlands have Rotterdam. Ireland is clearly a lot of food import and export, whereas some other countries are more self-supporting, so they don't import as much meat. However, if you buy, I just took a picture of a Tesco, but any supermarket you go to, you buy this southern fried chicken. If you buy a whole chicken, it says UK chicken, or if you buy beef, it says proudly UK beef. Has anybody ever actually tried to find out whether the chicken or beef or whatever you buy on your sandwich is also UK? It doesn't say it proudly, and it isn't. Most of the chicken on sandwiches will come from Thailand, South America. So food is actually being transported around the world. It's in transit for an amount of time. So if there's Listeria on it, that may even have a chance to grow. And any step you introduce in the process where it's handled, there's a potential problem of adulteration, of infection, contamination, paperwork going wrong. That's how the horse meat scandal was claimed to have started. You couldn't follow the paper trail at some point where the meat was coming from. And this is an international problem. Food, your prawns may come from Spain or wherever. Food is coming from everywhere, and we like getting more and more exotic food as well. So it has to come from somewhere. Now, I'm a scientist, and usually we get the question, we have to justify our existence. So what can science do about food poisoning? There's lots of different science. There's very applied science, that people developing equipment for helping companies. Our approach is that we try to find weaknesses in the bacterial pathogen, so the disease-causing organism, one that can be exploited for treatment, prevention, intervention. But then you can't just say, well, I'm just going to try A, B, C, and D, and hopefully one of these will work. Costs a lot of money, and you may still not get anything. So we try to go for a strategic approach, where you first need to learn about your enemy, and then find the weaknesses. That's a bit of a lot more long-term plan, of course. That's the scientist, so we try to help people. So what about the industry and the government? They need to do their bit as well, because they have the problem. For instance, improve production processes. Ensure that there's proper supervision, knowledge, good paper trails, good practices, that you don't get sloppiness in the factories where you can get contamination. Support innovation. I don't know whether people have tried this. This is the double bag chicken. So this is a chicken in an oven bag, and then the outside packaging. So you just remove the outside packaging, you put the whole chicken in the oven inside the bag, and only once it's fully cooked, you take it out and you open the bag. So you never have to touch raw chicken. Less chance of cross-contamination. You have absolutely no chance of washing your chicken that way. So that actually is I think it's a brilliant idea. But then the weakest link becomes the customer, you at home. Some people want to add some vegetables or want to have the juices for gravy or something like that. They want to put extra stuffing in the chicken, and they open the bag before it goes in. Defeats the purpose completely, so they still may touch the raw chicken. What you see here on the right is something that they're now introducing in chicken processing plants. One of the problems with chicken is that Campylobacter on the chicken meat doesn't like low temperatures, and freezing is actually quite good. However, you're not allowed to freeze the chicken because that reduces the value of the chicken, because we have a fresh chicken market. People wouldn't want to buy their chicken, frozen chicken, if they can have fresh chicken. So they need to find a way of getting the effect of freezing without actually freezing it. And they're doing different things. This is a cold nitrogen vapor that chills the surface of the chicken briefly to get the effect of the freezing without actually freezing the meat. They're also doing things, very clever things with what they call sonosteam, which is sonic behavior to open up the system and then get steam in to kill the bacteria. Very briefly, so you don't get cooking or anything of the meat. But again, the bacteria may be weakened and then survive less. Now, IFR is trying to help industry, regulators, consumers. So what we have now is what we call the Food Safety Center, Norwich, which has our research, has contact details, has information. Industry can contact us for support, assistance, information. Regulators can interact with us, but also people, consumers can contact us. Naturally, we can't say, is this chicken okay? That's not something we do. But we can give advice, or we can point people towards the right information sources. So if people are interested in reading about it, they can go to this foodsafety.ifr.ac.uk or contact people, and we'll try to help them. This is very important to get a central place where we can get interaction with industry and regulators to make sure that we, whatever we think of as scientists is also potentially introduced in factories or in shops. Other things we're doing. One of them is what we call typing or molecular epidemiology. Say the E. coli outbreak in Germany. How do you know whether all the E. colis are the same, or whether maybe 20 different cases of E. coli which were completely unrelated? We need to know whether it's the same case so that you have an epidemic, or whether they're all different cases. So what we try to do is try to find out as much as we can about the bacteria, and especially DNA data are relatively easy to get and very powerful for information, because you don't have to grow the bacteria necessarily. Where you want to try to find, for instance, is it always the same bacteria? So do you have a recurring troublemaker? Is it always the same bacteria coming back in the factory? Or has this case in Birmingham, is that the same as somewhere in Kent? Because somebody just traveled after eating the contaminated food. If you just have the geographic data, you may think, well, they're not related. But if you then find they have exactly the same Salmonella, there may be a link between them, and there's a reason why you should look further. And examples of that, where that is really successful, for instance, they did what we call whole genome sequencing. So the whole chromosome of the bacteria is rapidly sequenced. And for instance, TGAC on the Norwich Research Park is one of the institutes doing this kind of sequencing. This is one from the CDC in America, where they were very rapidly able to find that all the cases of Listeria they were looking at had to do with these caramelized apples, which are partially visible on the picture. But a more recent one, in the UK was in Newcastle at a food festival. There were cases of Salmonella, and it was a surprising different Salmonella than they would normally find, so they hadn't expected that one. And by finding out that all these people were related to this food festival, that everybody had gone there, and then had a dish which had curry leaves in there that were improperly washed and still had soil and everything associated with them. It was actually the Salmonella from these soil samples on the curry leaves that was causing the Salmonella outbreak. But by being able to get from everybody seeing the same strains, seeing that they all went to the same festival, by getting all this information, you then get an idea about where you're going and whether they're related or not. As I've mentioned, the E. coli outbreak in Germany, there's a bit more detail here. So it was in northern Germany, and I was actually in Germany when it was happening, and we were supposed to have a speaker on E. coli at the conference where I was at, and he was not allowed to come, because this was much more important. It was bloody diarrhea, serious complications, and what they call hemolytic uremic syndrome, HUS. And almost 4,000 people were affected, 53 died. And then you had the question, who done it? Well, the Spanish cucumbers, lots of economic damage, and it was bean sprouts that were responsible. What was surprising about it was the type of E. coli. The E. coli that normally causes these kind of problems is called O157. That's just the type of it. But this was an O104, which they hadn't expected. It was a really minor player in disease normally. So then they started looking at, so what happened with this E. coli O104? And they had a timeline of the original strain that they found in this completely unrelated case, this one-off case. And then they could, with the ones they found of the E. coli outbreak in Germany, they could actually do a sort of a detective analogy where they could find all kinds of events happening where they found out why this E. coli had suddenly become so dangerous when it wasn't before. It actually picked up a lot of tools or weapons on the way. So rather than being just a normal person, it suddenly became a G.I. Joe, which acquired things, lost things, and it became a lot more dangerous. And this can happen quite rapidly, because this was actually just a couple of years of evolution happening. So you could really go for the CSI microbes nowadays, where we're trying to, expand on this, where we use DNA information to track bacteria. Do we know where they are, where they come from? Do we have geographic information? Use DNA information to predict possible problems. Once we know these kind of things are happening, can we actually find out or predict what problems are going to occur? And that's where genome sequencing came up, as I say, at TGAC, the Genome Analysis Center on the Research Park. And this is an example of what you can do. In purple at the bottom is where the normal problem makers, the pathogenic E. coli, the O157s and so on were. And by making a sort of a tree where you group them into how related they are, you can actually see that the German outbreak strains, the red ones on the top, were very different from the standard problem makers. So there's a new problem occurring. And the ones in between are all E. coli from the lab and so on that are not causing disease. So they are really different. We can also do other ways of predicting things. This is something called Combase, which is assisting regulators in industry and is one of the high-impact things that IFR has helped develop. Basically what this is a really large database of information on how bacteria grow and survive in different conditions, in different food sources, in different media, any information. And when you have these three lines like the 5 degrees, the 10 degrees, and the 15 degrees, if you have that information, you can also start predicting what will happen at 12 degrees or 8 degrees, so information you don't have. Now, why is that important? It actually gives an opportunity for industry and for regulators to sort of test on the computer in silico what's going to happen if they would change the production process. Can they take that risk or not? And the outcome of that may save a lot of money, but instead of just trying it and hoping that you don't get disease, you may actually already predict this is not going to work. You may get more Listeria survival, so your product will become more dangerous. Relatively simple, the idea. Of course, it's very much complicated behind the, just showing the graphs, but really helpful to the industry and to the regulators. Same here, this is how Clostridium spores can germinate, so when they start growing again and become bacteria. If you take 100 spores which are exactly the same and you put them in exactly the same conditions, they will have a lot of variability. So some of them will start growing very early, some other ones will start growing very late. If you know the variability, you can model that in a mathematical model, you can then start predicting what are the risks I'm taking. So with this kind of information, you could do risk assessments. And I always like this picture with the cat. I'm a cat person, sorry. Is it worth it to get the bird? Sometimes it's not worth it. And the other one I really like to say, well, what can a risk manager tell you? Be careful. So finally, a little bit about Campylobacter to wind up. Campylobacter said most common cause of food poisoning. So we had this one already, say the risk of developing paralysis. People sometimes say, well, what about organic or free-range chicken? Is that better? No, it's not. It's actually found probably more likely to be organic chicken. So whether you have factory chicken, organic chicken, free-range, they're all the same. What is this paralytic syndrome? Well, this is called Guillain-Barre syndrome, after two people who described it first. It's basically people eat chicken, get Campylobacter food poisoning, and possibly weeks or months later, they start developing paralysis syndrome. So it starts with a tingling sensation, some pain, their reflexes become worse. At some point, they can't work anymore. They may even need ventilation. And there's a lot of healthcare, and there's actually a video from the Food Standard Agency, which anybody can watch, the truth about Campylobacter. And this person that's in the video had Guillain-Barre syndrome. And she was in a wheelchair for a few years, and she will always need a stick to walk now. So it has improved a little bit, but she has lifelong problems from this one Campylobacter food poisoning case. And what happens is you see here, on the top you see a nerve sheet. And on the nerves there are certain sugar groups that are on what they call the myelin, which is the cover of the nerves, which is very important. Campylobacters have learned the trick of making sugar groups that look like this myelin sugar groups. So in the people who have Guillain-Barre syndrome, they had the infection, their body starts making antibodies against the Campylobacter. These should normally be not produced, because these antibodies start attacking your own nerves. So the Campylobacter, in a way, didn't even do it. It just induced a process where you're attacking your own nerves. So it's an autoimmune disease. But very important, very high morbidity cases, people needing ventilation and everything, and lifelong problems afterwards. Just from a bit of undercooked chicken. Now, there is a lot more public awareness now of Campylobacter. So these are clippings from November last year, where Asda got a bit of a hit, worst for poisoned chicken, supermarkets sell contaminated chicken, how dare they? Food poison bug spread by workers at chicken farms, examples of people who had Campylobacter. And why was that? The FSA decided that nothing is working. Prices of chicken have dropped significantly, which means there's very little space for developing new technologies to reduce Campylobacter on chicken. So they wanted to put pressure on the retail sector, and they actually started most of us call it naming and shaming. The FSA does not like that one. They say it's naming and praising, because the ones who do best are praised, they think. Well, I don't know, but the winner, if you can call it a winner, Tesco had only 64% of their chicken had Campylobacter at retail. So this is whole chickens taken from the shops. So 2/3 of the chicken still had Campylobacter on it. Depending on which one you're going, 11% was the lowest had the highest level of contamination on the outside of the chicken. And even there were packs on the outside contaminated. So you could imagine you take your chicken, you put it in your trolley, and then you start licking your fingers. You may get Campylobacter that way. Now, again, I don't understand why people would do that, but it's possible. As you can see, there was virtually no difference, 64%, 78%. It means that 2 out of 3 or more chicken are contaminated with Campylobacter. That's incredibly high. Number of samples were based on the market share they had in 2010. So this is why we quite often get the question, why are Aldi and Lidl? Well, in 2010, they had a very small market share. The FSA is going to do a second year of testing, and then Aldi and Lidl are included as separate supermarkets, because now they have a big market share. So we should stop it. It's easier said than done. Already in the 90s, when I was working less, I was working on Campylobacter, and the problem is still as big as it is now. So we haven't been particularly successful yet. Campylobacter you can get from a lot of sources. So as I say, at the top you see cattle, pigs, chickens, sheep, wild birds, dogs, cats, person-to-person transmission, water, milk, loads of different opportunities to get Campylobacter. We basically, if you simplify the problem, it's somewhere in the beginning of the food chain. It gets onto your food, we eat it, it gets into you, and you get disease. So there's 3 places where you can stop it. Stop it getting into the food chain at all, so get rid of it from the chickens. Get rid of it surviving in the processing plant, or make sure that people cook their meat properly, and so the Campylobacter is dead. So what we've been doing is mostly looking on the survival in the food chain and on the bacteria itself. So what can we learn about the bacteria to find these weaknesses? And this is one example of what we're trying to do. What you see at the top is a picture of your intestinal lining. On the intestinal lining there is a slime layer to protect the cells. And then your food is in that light blue at the top. Now the Campylobacters come into your intestine with the food. They don't like it in the food, they want to get towards these intestinal lining, so they swim towards the intestinal lining through the slime layer, and then they're getting into the cells or even through the layer. So they need to know where they're going. Bacteria as much need a sat-nav, because you can't just go anywhere and just hope that you get to the right destination. So bacteria also have a very directional movement. And what they do is a sort of a smell nav. They smell chemicals. So there's some chemicals they like, and our noses in a way has developed the same way. Sometimes you smell the bacon and you go towards the bacon or the croissants or something, or you smell some really foul odor and you run away from it. In principle, that's what bacteria do as well. They smell the environment and move towards or away. So we've been trying to interfere with that process, and this is actually pretty much the only time I show some real lab stuff. What you see here is bacteria, and this is a tube with an agar, which is a soft agar where the bacteria can go onto and can swim through if necessary. And where you see red, that's where the bacteria are. So we started them at the top, and we just allowed them to swim into the tube. As you can see, the left one is what we call the wild-type bacteria, where we haven't done anything to. And it swims about 2/3 of the tube. Then we started inactivating some of the genes in it. As you can see, when we inactivated the first 4 genes, they don't swim as far anymore. So somehow we confused them into not swimming as deep as they normally go. The next one, which says 1110C, with the yellow arrow, that one is actually differentially confused. It's now swimming further. So somehow it's thinking it's nicer there. It's almost like some people can't smell hydrogen sulfide or other bad smells, and they would walk towards something that other people would stay away from. So we have now one where we said, okay, this one goes too far. And then we started combining them to see which one is sort of a dominant one, so which either not going far or going further is more dominant. So what is more important, the nice smell or the bad smell, if you get a mixture of them? And we can see that some of these actually, the one that goes too far is the non-dominant one, because if we combine that one, it can't make the bacteria swim further anymore. And the way we try to, this is probably the best way of showing that, is that the bacteria has like the 2 sat-navs on there. One of them says veggies and you need to go towards the veggies and that's good for you. And the other one says, oh no, you need to go to the cake and the French fries, they're much nicer. For bacteria, this is a very important, do you stay somewhere where there's food or do you need to go elsewhere where there's food? So the idea now is that we know that we can interfere with them by inactivating stuff. Can we now feed them something that they would move away, for instance, from the chicken skin, or they would go deeper into the chicken skin so you don't easily get contamination? This is not easy, but it's just an idea of which direction we could go. A similar one is, how do they survive outside, on the chicken skin or in factories? Because we know they do, but if we take Campylobacter in the lab, it's very easy to kill. Put it on a chicken skin and suddenly it's this real mean survivor. So what is the difference between this chicken skin and our laboratory? And one of the things we've been trying to do is what we call a chicken juice. This is freezing a chicken, a whole chicken. You put it in the beaker. This is an actual experiment. And you just let it defrost. And you see this red liquid, the runoff from the defrosting chicken. You gather that red liquid, you filter it to make sure there's no bacteria in it anymore. And that's what we call biofouling material. So what you see at the top is what we call a biofilm. It's where the bacteria attach to a surface, and then they start covering themselves with a slime layer, which protects them against disinfectants, antibiotics, and all things they don't like. And at some point they leave that biofilm again. And what we did here with the chicken juice is the one which is set A on the side is just bacteria on a surface with an electron microscope. So all these little rods here are single bacteria. And you can see it's a clean surface and the bacteria are sort of spread all over this surface. Now if we add 5% of this chicken juice, of this organic material, I hope it's visible, but suddenly this surface is not clean anymore. It has sort of a layer of dirt on it. And the bacteria start grouping together, clumping together, and there's all kind of material in between the bacteria. And if we add, the undiluted chicken juice, suddenly you see a lot more bacteria and the whole surface is covered. And we know now that these bacteria actually are very happy. Normally they would die, but in these conditions they're happy and they're surviving. So this chicken juice seems to make them better at doing that. And there will be a lot of this organic material in factories and everything, so this may be one of the things that gives Campylobacter the chance to survive. Can we do something about it? Yes. Now this is again very much in an early development, but we've been doing work in my lab where we started working with enzymes, trying to break down, if we go, so this material, this gunk that you see between the bacteria, it contains DNA and several other things. And we know now that if we add certain enzymes like DNase, which is an enzyme that degrades DNA, you can see on the first one on the graph is just the bacteria on this clean surface. The next one is with the 5% chicken juice, so you get more bacteria surviving and more material being gathered. If we then add the DNase enzyme, we seem to be able to get it down to almost the standard levels. And DNase is already being used clinically. This is pulmozyme, is a DNase enzyme. That's people with COPD, pulmonary disease. They have a lot of slime in their lungs, and this is an enzyme they use to break down the slime. So this could even be is already clinically used. It's currently way too expensive to use on the factory floor. We're working with a local poultry producer where we're interested in see whether we can do something about getting this more applicable on the factory level. And in the end, the idea would be, can we make novel cleaning regimens that actually get rid of the Campylobacters? And finally, what can you do about it? We've worked with a show called Watchdog Test House, which is daytime television. They asked us whether we could do something on Campylobacter. And we were in a show together with how much mileage you get with your car. So it's just a consumer show that combines anything from rechargeable batteries to cat litter and also Campylobacter. And what we did with them was just to show that we can do something about it in your house as well. So we bought some chicken, standard supermarket chicken. We sliced it up, and we grew some Campylobacter. So the second picture on the right, on the top, a high concentration of Campylobacter, medium concentration, and no Campylobacter. And what we're doing there on the bottom big picture is that we're dipping them in either the high, low, or no Campylobacter solution. And then one of these slices went into the fridge for an hour, one of the slices went into the freezer for an hour, and one of the slices went into the oven for 20 minutes. And then what we did is we took the chicken, took all the bacteria off, and just counted how many Campylobacters were surviving on that chicken. And this is an example at the bottom here. The H is for the high concentration, the L is for the low, and the 0 is for no Campylobacter. The left one is the freezer. You see, this is a dilution series, so the further they go down, the more surviving there are. Loads of bacteria of the high one, some bacteria of the low, nothing on the 0. In the fridge, they survived fine as well. The cooking, completely gone. So that piece of chicken, once it was cooked in the oven, it may have been a bit dry, but it would have been fine to eat. Now, it's a laboratory, so we're not allowed to eat in there, but that chicken would have been safe for consumption. So, it's a big problem, it's an important problem, but when I was asked about, so who's to blame, I gave the perfect politician's answer, said it's not about who's to blame, it's how we can solve the problem together. And everybody can contribute. So this is from the Russell Howard Good News Show. It's not all doom and gloom. The industry needs to do their bit. Regulators need to make sure that they support the industry in making food still affordable and safe. And you at home need to make sure that you're aware of what you're doing and properly prepare it. And don't leave it too long. Don't go past the sell-by date or the use-by date if you're not sure. So proper food handling, proper food storage, proper food preparation. Don't do what the TV shows are, because what they make in there you wouldn't want to eat. It's only for TV, and TV is not real. And we need to get as safe a food as we can, but make sure that you clean properly, that you, and say, my kitchen, I'm not practicing what I'm preaching, unfortunately, myself as well. I'll be fair enough, my kitchen is not a good example. But we can actually have no real problem with food poisoning if we know what we're doing and we're preparing the food right. In a restaurant, of course, go look at the foods, the star rating. Go to the 4- and 5-star restaurants, not to the ones with 1 star. Don't go to that Nottingham kebab shop where they found fecal matter, mouse droppings and everything in the meat. That one would never get a 4 or 5 star when they would be tested. So check the food hygiene rating if you go to a restaurant. It's not a certainty, but it reduces the chances of food poisoning. So with that, I want to stop and thank a few people, the IFR Comms team, for organizing this, the people from here who are supporting us, the audio and so on. We do get our funding. I mean, we can't do anything without funding. We do get funding from the BBSC, the Biotechnology and Biological Sciences Research Council, very important. The Food Standard Agency, from industry and all kind of other sources. It's something that we do together. It's not like it's all individuals working on it. It's a big team of people all interested in different aspects of food safety. And I hope I've been able to do the right-hand one and not the full right-hand one where you're all fallen asleep by now. And with that, I want to stop and thank you for your attention.
Thank you, Arnoud.
Anybody hungry?
As you can see, the scientists in the Food Safety Centre may make good scientists, but they do make very poor dinner dates. Best to have them
good after-dinner speakers.
Yeah, after-dinner speakers, possibly. So have we got any questions? Any questions from the audience?
Hiya. You mentioned earlier about the kind of
Sorry,
sonic
the lights
steam.
are very bright, so...
Oh,
yeah,
sorry.
Yeah, go for it.
Yeah. Could you just explain a little bit more of how that works?
Sorry, which one?
The steaming of the chicken.
Yeah, so Campylobacter and other bacteria don't like steam. The problem, if you steam it, you can imagine you very quickly cook the meat or the surface of the meat. So they have to find a way of getting the steam to go through the surface of the meat without cooking it. So it has to be a really short burst of steam, so you don't get cooking, but it still has the effect. And somehow the sonic part of it is almost like opening pores in the meat, allows the steam to better get through and get out again without cooking the meat. as that's how they've told it to us at conferences. It's a proprietary process, of course, so they won't give us the exact details. But it's one way of doing it. I mean, people are trying all kinds of things. They're trying to change the feed of the chickens. And they've tried what they call essential oils. The problem is you don't want to get the meat already getting a certain taste or color. You don't want the meat that you buy your normal chicken that already has a bit of a garlicky taste. So again, they need to find it in such a way that consumers will still buy it. So anything that we do that changes the appearance of the meat will have a negative effect on sales, which defeats the purpose then, at least from the industry point of view.
Any more questions?
There's one, yeah.
Good evening. I grow organic vegetables.
Mm-hmm.
So about November, I have a couple of tons of cow manure delivered. I dig that into my soil, and then I plant my potatoes and my brassicas. Am I taking a huge risk?
Nothing is risk-free. Again, many of this, it's not a problem if the manure and everything, if there are bacteria on there, but then you need to properly prepare it and wash it and everything. So if your procedures are right, you're not taking a big risk. I'm not saying you're not taking any risk, but then again, eating anything can be a risk. But again, it's all about how clean you can do it and how properly you can wash it and everything to when you prepare it. Raw vegetables are a bit more risky than cooked vegetables. But there's nothing we can do about that. If you would boil your salad, it's not going to be particularly nice.
There's one just down here, Matt. Two down here, Matt.
Hello. Is one reason that we have these highly pathogenic bacteria around due to the fact that we are suppressing the more benign bacteria, which would be competing with them on, say, the surface of the chicken?
Not really, I think. A lot of disease is also to do with us. I mean, the bacteria may have not completely changed, but we are less exposed. I mean, I don't know whether you've heard of the hygiene hypothesis. This is this idea that we get more and more allergies and so on nowadays because our children don't play in the dirt anymore, so they don't get exposed to bad bacteria and they don't learn to recognize them. Our food consumption pattern does change our gut bacteria. So junk food has an effect on your gut bacteria, which makes you more susceptible to infections potentially. So I don't think it's as simple as that we're suppressing the good bacteria. Bacteria are smart. Anything we throw at them, they'll come back with something. It's an arms race. So this is where antibiotic resistance comes in. When penicillin was found, they thought it was a solution to everything. In the beginning, antibiotics were thrown at everything. Even, people go to their GP, they want to have a pill, they want to have something done, otherwise they don't get the feeling the GP is taking them seriously, so you give antibiotics. And now we are having the problem that comes with that. We have been using so many antibiotics that we get the resistance against it. Some countries like the US, you can buy certain antibiotics just at a gas station. That's lunacy. It's really bad. And you get more and more antibiotic-resistant bacteria there. Are these bacteria more pathogenic? Not necessarily. But they are learning tricks. And so I think it's a combination of all of this. We have maybe less good bacteria in our gut, less protection. We have more pathogenic bacteria picking up tricks. Because we have so much food going around the world and so much different food interacting with each other and so many bacteria having a chance to interact, they learn from each other as well. They pick up what we call genetic traits. So I can't make it any nicer than it is, unfortunately. I'm not sure whether there are good or really bad bacteria. It's just you have good bacteria when they're put in a certain situation become bad bacteria. That's what they say to the judge, at least.
Hello.
Yep.
In the poultry industry, they use vaccinations quite a lot because my partner actually works in that industry. Mm-hmm. What do you think about the use of vaccinations to tackle things like Campylobacter?
They have tried vaccination for Campylobacter and have been unsuccessful. One of the problems, again, is that bacteria circumvent any measures we're taking. So when you start doing a vaccination against Campylobacter, what you'll actually get is bacteria that don't express the thing anymore that your vaccine is working against. So if you make it against, say, a surface protein, you will select for the ones that don't have that surface protein, and they're very variable. And not just Campylobacter. I know, for instance, that against streptococci, pneumoniae, where they have, against the 7 most common serotypes, they have vaccine now. And that's very successful. But in Rotterdam, in the Netherlands, they have done this, they're doing this huge project where they're following children already for 10 or 15 years. They're following that whole generation. And what they found was that by vaccinating against these most common serotypes, they're getting rid of these 7 serotypes. What they're getting now is all kind of other bacteria. The streptococci aren't there anymore, so they move into that niche. And you may actually be worse off. So the Campylobacter vaccine is problematic because it doesn't work very well yet, and we don't know what we're getting in return from it. So it may in the end be better the devil you know and just prepare our food better. But that's not the message. Consumers rightfully can say it shouldn't be there in the first place. But we're talking about a bacteria that has grown in birds millions of years now. It's been very good at what it's doing. And we're trying in a few years to fight millions of years of evolution. We may be a little bit too impatient.
Arnoud, you had the data on the contamination of chickens in the supermarkets.
Yeah.
What's the level of contamination of Campylobacter in the chickens going into processing?
It's about the same. What they see is that's one of the things they're doing as well, because again, they need to change procedures. There's something called finning. Maybe you've heard about it if you... Basically what happens is they have a certain amount of space for the chickens, so they put the chicks into the barn and they start growing, but they want to use that space as efficient as possibly. So they're normally grown until day 39 or 40, but they put so many chicks in that space that already the space is taken up at about day 30. What they then do is they go into the barn, a quarter of the chickens is taken out, so that the other 3/4 have more space to grow. Normally they put in a lot of biosecurity to ensure that Campylobacter doesn't get in. And this is of course an enormous breach of that security because people have to go from farm to take these chickens out. And this is when you start seeing Campylobacter becoming detectable. So for instance, M&S is now doing what they call a no-finning policy, which means that the space that is there can be used for less chickens. However, that means that the price per chicken is potentially getting higher. So maybe we have to pay a little bit more for our chicken for safety. Which again is not a message that is particularly well received usually, but we may not get it as cheap as we wanted.
We import a lot of our food, and I guess that the supermarkets, the buyers, work with international producers who are probably subject to national government regulations, but are there any global regulations that are being developed? And I realize it would be a minimum standard.
In principle, food that's being imported in the UK will have to conform to UK regulations and paperwork. Although, again, that's not my area of expertise. You can't just import something and say, well, in my country this would have been fine if you want to import into the UK. The problem, of course, is do you get grey imports? How well is it checked up? Because the FSA is not an enormous department or whoever has to check it. So you will get things getting through that you would have not liked. And this is where the horse meat scandal was a very good example. At some point in the trail, the data got lost, and then suddenly it was relabeled from horse, it was suddenly relabeled beef. So this is again why you need checkups as well, to make sure that not just the regulations are good, but also that they're adhered to. And this is why, and I say the horse meat scandal was a very good example of where safety was not an issue. The meat was perfectly safe. Okay, I know people don't want to eat horse meat, but there was no danger to the public. But it erodes consumer trust, because if you can't trust what's on the label, you don't like it. and you're not buying it anymore. Because if it says on the label it's beef, it should be beef and not beef with horse. Not a safety, in this case at least, it was not a safety issue. It could have been if there would have been hormones in there. But again, the paper trail was insufficient at some point, and that should have never been allowed into the country. And this is where regulations, if they're adhered to, they're fine. They're not adhered to, we have a problem.
Thank you. Can I just ask you, when we buy fruit, vegetables, salad leaves, we tend to take them home and quickly wash them in cold water, running water.
Mm-hmm.
Now, what does that do to the bacterial content?
In principle, not much. It's just washing it. It's like washing your apple. It's not necessary. It should already be clean when it's sold in the supermarkets. But we do it. A lot of people still do it.
Can I just add then that I think one of the most interesting things you've told us, amongst many tonight, was the number of packs of chicken that are contaminated on the outside. So when I pass that through the supermarket checkout, and it's put in the basket with a lot of stuff I'm going to eat uncooked. Mm-hmm. So, all the things you've said about cross-contamination are really undone, aren't they? And
yeah, the only thing I need to clarify on that one, although there was a significant percentage had Campylobacter on the outside, only I think 3 or 7, I can't remember which of the two it was, had actually the high level of Campylobacter, which would be the big risk. So the chance is quite small, but it's there. I have a news clip on a slide somewhere where it said, just touching the packaging made me ill. Unfortunately, it was from the Daily Mail, so how trustworthy it was. I think, yeah, that was behind you, Mark. Yeah.
Considering, sorry, hello. Considering Campylobacter manages to survive the entirety of the food production process, why is it so difficult to grow in the lab?
Because we're not doing it right in the lab. Now, somehow it seems to be that the environment is on chicken skin. There seems to be something on chicken skin that suddenly helps it to survive a lot. But remember, in the lab we try to grow it. So growth in a liquid medium is very much unlike the real-life situation in a gut or on a chicken skin. In the lab, we have different goals for what we're doing when we're growing bacteria. So these bacteria have been growing fast. They're quite often already what we call lab islets, so sort of adapted to the life in the lab. So they may not be as representative anymore as the outside bacteria. So it's just that our conditions have different purposes. So, but yeah, it's one of what we call the Campylobacter conundrum. Difficult in the lab, survives outside without any problem. So we're the problem then.
Okay, well, I think that's probably time to wrap this up. I'd like to thank you all for coming tonight to the talk, and please look out for further events from the Institute. We would welcome you to those, and it just remains me to thank Arnoud van Vliet one more time. Thank you, Arnoud.